eQTLs are genomic variants that affect the expression of genes, and we can distinguish two types of eQTLs: cis-eQTLs, which affect the expression of genes nearby, and trans-eQTLs, which affect the expression of genes farther away. Although cis-eQTLs play an important role in gene regulation, they only account for 30% of gene expression variation, while trans-eQTLs are estimated to contribute the remaining 70%. The discovery of cis-eQTLs is quite straightforward. Because they often have large effect sizes, it's easy to find them by simple linear regression of a SNP's genotypes and the target gene expression. And because only genes in the vicinity are considered, there are only a few SNP gene pairs to test. On the other hand, trans-eQTLs have typically small effect sizes, making them hard to detect. In our approach, we reverse the direction of the regression by explaining the SNP dosage using multiple linear regression on all genes. In this way, we achieve two goals. First, we can aggregate information from all genes, and second, we avoid the multiple testing burden of testing all SNP gene pairs. It's important to note that for each SNP, we remove all the cis genes from the explanatory variables to avoid discovering cis-eQTLs. Reverse regression tackles all this problem and is implemented in our packet called TEGES. We use simulations to show that it has increased power to detect trans-eQTLs, obtaining a higher area under the ROC curve compared to traditional methods. Different simulation settings show that we can gain more power for trans-eQTLs with many target genes and with bigger effect sizes. In real data, we run TEGES on all GTEx tissues and show that it predicts more trans-eQTLs than expected by chance. In these two examples, the Manhattan plots indicate that significant predictions can be found throughout the genome, where each colored dot is a genome-wide significant trans-eQTLs. If we plot the genomic coordinates of the trans-eQTLs versus their target gene, we can observe that each trans-eQTL targets many genes, even in other chromosomes. Our predictions also support the idea that trans-eQTLs are tissue-specific. Even after removing 4 SNPs in linkage disequilibrium, 70% of them are discovered in single tissues. Moreover, many trans-eQTLs overlap with previously discovered cis-eQTLs, even though their cis genes have been removed by our approach. This indicates a possible mechanism of action via cis genes. Further analysis support our results with trans-eQTLs being enriched in functional annotations. They are more likely to occur within enhancer regions and less likely to occur in heterochromatin regions. Statistically significant enrichments are also found across GWAS traits. We expect these findings could help us understand better the genetic architecture of complex traits and diseases. If you would like to know more, check out our bioRxiv preprint.